Welcome back, students. In this session, we'll discuss fertilization and implantation. So, you already know that in human reproduction completes in 6 reproductive events: gametogenesis, insemination, fertilization, implantation, gestation, and parturition. So, let us start with the fertilization and implantation. The question arises, what is fertilization? So, fertilization is a process of fusion of haploid male and female gamete that results in formation of diploid zygote. And you remember, diploid zygote is a totipotent cell. Why? Because it has a capacity to develop into new individual. The zygote has a capacity to develop into new individual. So, in case of animals, zygote is a totipotent cell, whereas in case of plants, all plant cells are totipotent. They have a capacity to develop into a new individual. So, let us start the fertilization. So, soon after the fertilization in case of human beings, it takes place at the upper part of the fallopian tube or at the junction of ampulla and isthmus. So, soon after the sexual intercourse, the semen is released into the female genital tract by the process called insemination. Soon after insemination, the activation of sperms takes place by the process called capacitization. So what is capacitization? On the sperm head, you find a fatty layer. That fatty layer is removed by the content which are present in the genital secretion. So after capacitization, the sperms acquire the fertilizing capacity, or they acquire capacity to fertilize with the female gamete. So with the help of a vibratile movement of tail, the sperm reaches the upper part of the fallopian tube. So look at here. Soon after ovulation, secondary oocyte is released into the fallopian tube or oviduct. With the help of ciliated epithelium, the female gamete, being non-motile, it moves towards the upper part of the fallopian tube, where the fertilization takes place. That's why in case of human beings, the fertilization is internal. So it takes place in the ampulla isthmic junction. So soon after ovulation, it reaches the upper part of the fallopian tube within 6 hours. Simultaneously, sperms are transported to the upper part of the fallopian tube. Look at here. So insemination, the activation of sperms by the viscous fluid of the fallopian tube, that is we call it as capacitization. Soon after this, when sperm establish the contact with the female gamete, female gamete will produce a glycoprotein or hormone-like chemical substances called fertilizin. That fertilizin which attracts the male gametes towards the female gamete. As a result of release of fertilizin, the movement of the male gamete increases. Or motility of the male gamete increases towards the female gamete. Once the sperm establish the contact with the female gamete, the sperm releases anti-fertilizin, so which neutralize the effect of fertilizin, so that no more sperms are attracted towards the ovum. This reaction we call it as a fertilizin and anti-fertilizin reaction, and it is a highly species specific. Next, so soon after the sperm establish the contact with the female gamete, the sperm head releases, two enzymes, proteolytic enzyme, that is acrosome releases proteolytic enzyme or hydrolytic enzyme. One is okay hyaluronidase enzyme. So this hyaluronidase enzyme which dissolves corona radiata, then the acrosome releases one more enzyme called acrosin or zona lysine, which dissolves zona pellucida and makes microscopic passage. Through this passage, the sperm enters into the female gamete. Female gamete. So look at here. Once the sperm establishes the contact with the female gamete, immediately the female gamete, that is secondary oocytes, completes second meiotic division. As a result of this, one ooted, one functional ooted and two non-functional polar bodies are produced. And immediately the polar bodies, which does not play any role in the process of fertilization, immediately they are produced and they are get degenerated. So look at here. Soon after the penetration of sperms, okay, the uterine membrane becomes thick, and this thick membrane we called as a fertilization membrane, which prevents polyspermy. The male pronuclei, okay, move towards the female pronuclei. As a result of this, both the pronuclei, male and female pronuclei, fuses and to form a diploid zygote, and this process we called as a amphimixis or karyogamy, or this we called as a woman has conceived. Is that it? Then move on to the next part, the significance of fertilization. So fertilization, as a result of fertilization, the oogenesis process is completed. So in the absence of fertilization, oogenesis process is incomplete. Second one, it determines the sex of the fetus, male or female, and restoration of chromosomal number takes place. Before the formation of gametes, they are diploid in nature. After the formation of gametes, they are okay, they are haploid in nature. After fertilization, again they become diploid. Diploid. Then we'll move to the next function. That is soon after fertilization. Soon after fertilization, that initiates the developmental stages, or it introduces centrioles in the womb that brings genetic, okay, centrioles in the womb that initiates the embryonic development. Out of this, the fertilization brings genetic recombination that leads to variations and evolution. Next, we'll move to the embryonic development. Soon after fertilization, the diploid zygote enters into the three developmental stages. One is cleavage, second one is blastula, third one is okay. As soon after formation of blastula, it get implanted into the uterine endometrium by the process called implantation, and after implantation, it enters into the gastrulation. So let us start with the cleavage. The question arises, what is cleavage? The repeated mitotic division of diploid zygote that results in the formation of multicellular morula is called cleavage. Look at here. Soon after the fertilization, zygote enters into the repeated mitotic division. As a result of this, single-celled zygote is converted into two-cell stage. Two-cell stage is converted into four-cell stage. Four-cell stage is converted into 16-cell stage, or we call it as early morula, and 16-cell stage is converted into 32-cell stage, and that is we called as a morula. So mitotic division of single-celled zygote to form 16-celled morula, and this process we called as a cleavage. And one more thing, very interesting uh, aspect is during the process of cleavage, the cells keep on dividing. The size of the size of the embryo remains as it is. Remains as it is. Next, look at here how exactly the cleavage takes place. How the zygote is transformed into 16 cell stage or morula. The egg divides completely without interference of yolk. This is we called as a holoblastic division, holoblastic equal type of division, or complete type, because the entire cell divides, that is called holoblastic. If incomplete division takes place, that is called meroblastic cleavage. In case of okay human development, embryonic development, the cleavage is complete, holoblastic, equal. So zygote, the cleavage takes place in the fallopian tube. Soon after the fertilization, that is 30 hours after the fertilization, okay, the zona pellucida persists, and first division is vertical division. Okay, the furrow appears at the animal pole, continues up to the vestral pole. As a result of this, single-celled zygote is converted into two-cell stage, and two daughter cells are produced, and these daughter cells are called blastomeres. So second cleavage is also vertical but right angles to the first one, and it occurs 60 hours after the fertilization. And second cleavage is vertical but perpendicular to the first one, or right angles to the first one. As a result of this, two cell stage is converted into four celled stage, or as a result of this, four daughter cells are produced, or four blastomeres are produced. Look at here, these are the four blastomeres. And third cleavage is a horizontal takes place. Okay, altitudinal division takes place, or horizontal division takes place, and it takes place 72 hours after the fertilization. So 4 cell stage is converted into 16 cell stage. This we called as a third cleavage, or is horizontal or latitudinal. The 4 blastomeres divide horizontally to form 8 blastomeres. Fourth cleavage, so 8 cell stage is converted into 16 cell stage called morula, multicellular morula. Okay, multicellular morula. And you remember, the multicellular morula is an intermediate stage between cleavage and blastula. So advanced morula, the outer small cells are we called as a transparent cells are called micromeres, and the inner larger cells are called megameres or macromeres. So look at here, morula reaches the uterus in about 4 to 6 days, and morula now enters the uterus and start absorb uterine milk or fluid, and that is not sufficient. So soon after this, what happens? Okay, implantation process takes place. Implantation process takes place. So zona pellucida, the protective egg membrane, still intact with the intact to prevent implantation at the fallopian tube. So instead of uterine endometrium, if implantation takes place in the fallopian tube, and such a type of pregnancy we called as a ectopic pregnancy or tubal pregnancy. So look at here. So here implantation takes place within the fallopian tube. Then 16 cell human embryo on a pin 4 days after the FERT, this look like this, 16 cell stage. Each blastomeres has a capacity to uh, form a complete embryo. So morula has a totipotent cell, and it has a capacity to develops into new individual. And you remember one thing, in case of human beings, the cleavage is indeterminate, because the fate of the embryo is not determined at the early stages. If the fate of the embryo is determined at early stages of the embryonic development, and such type of cleavage we called as a determinate cleavage, okay, whereas a human female, okay, the development takes place, or cleavage takes place in indeterminate manner. So soon after the formation of morula, so morula is transformed. Multicellular morula is transformed into single layer blastula by the process called blastulation. By the process called blastulation. The outer layer of morula start absorbing uterine milk, forms a layer called trophoblast cells, and blastomeres rearrange, and a new cavity is formed. That cavity is called a blastocele cavity. So when you look at here, this is a blastocele cavity, and during the process of blastulation, what happens, the cells start to move towards the periphery and arranged in a single layer. As a result of the movement of cells, a cavity is formed, and that cavity we called as a blastocele cavity. Okay, so this completes the fertilization process and early embryonic development. In the next class, we'll discuss implantation. Thank you.